sorry about that. Something happened with my computer, so I had to restart. Okay, let me load back my presentation here really quickly. Okay, so again, what I was saying was, okay, so I wanted to take some time today and review some of the topics that we discussed during this program. As you remember, we talked about how the ability to sequence viral genomes rapidly allows us to explore what is going on in almost real time. And we have this abundant data that we can explore and study. And this data is always updated and allows us to really understand how the virus evolved over time. But also, we have access to other types of information that actually tell us a lot about the host response. And so not only do we see an exponential growth in the different types of viral and metagenomic data associated with COVID-19, but we also see host response, environments, and how these different elements interact using these high throughput technologies like next generation sequencing. So we learned about the major aspect of connecting the dots between biological samples and what is in the data, speaking about different types of sequencing techniques, where we looked at targeted total DNA and RNA, and also amplicon and environmental targeting. And these techniques really allow us to study how the virus changes, identifying mutations. It helps us identify presence or absence of specific viral strains or viral sequences, and also allows us to study the differences in composition when the virus is in other types of environments. So we talked about how we can detect specific strains of viruses. We talked about how we can look at different types of environmental samples and find different composition elements like taxonomy and variability. And what's important to understand is that a lot of this information is actually hidden sort of in this unstructured data that we have to deal with, right? So it's very important for us to understand what types of data contain what types of information. We talked about FASTQ data, which of course comes kind of like as the raw output from sequencing. And many times those reads could be aligned to different types of references that will explain where these reads fall on what specific genome. We of course looked at a lot of FASTA sequences of assembled genomes, and those sequences are representative of some kind of a consensus that we have in a given biological sample. And we also looked at the role of annotation. So annotated versus unannotated genomes really are the, give us the ability to interpret what's going on, right? So when we look at the process of alignment of reads, we have to understand how these reads are representing what is going on inside these biological samples, and different parameters about these reads and about the files that contain the reads actually give us more or less information about what's going on. So for example, we can look at different quality of reads. We can look at how these different reads are arranged together to form pairs, or whether they are spread out in longer segments or condensed in shorter segments, which really allows us to understand how the specific sequences that we have in real data can be or cannot be interpreted, right? And so we had a few different examples that show us how quality of reads or mapping quality on a specific organism can help us understand whether there truly is a presence of a specific virus or a specific sequence in our sample. In application to COVID-19, these types of insights are very important as we continue and build our interpretation or our understanding of what's going on and the data that is being generated that kind of informs this understanding. So we also looked at, many different examples of data sets being submitted very rapidly as the virus evolves, and the types of possibility that that opens for us to understand variability in terms of evolution over time, gives us understanding of potential origins, also kind of gives us insight into how the different populations are affected by this virus. So, I know that there have been several questions that have been posted on the different forums. So I just want to take a little bit of time and go to the program pages. So here, I've been noticing that there has been a lot of activity. And I wanted to point out that, many of the pipelines, right? So a lot of the questions that I saw was that we have, some kind of slowing down of the processing speed in terms of how fast these pipelines run. So I wanted to just kind of point out why that is happening. So right now on the platform, we have many pipelines that are running, right? Now I'll show you guys the back end just that you can see what's going on. So when I go to the back end, I'm able to see how many pipelines are running. And so I think this will kind of show you what is going on and why things are slowing down. The way that the server is organized is it actually runs parallel processes on a number of CPU units assigned to each process. So you can see that these are all of the pipelines that are being processed, right? So you can kind of go through and, you keep going and all of them are still processing, which means that they are, currently utilizing the CPUs that we have available. Now we have a limited number of CPUs allocated towards each process. And okay, so you can see how many up until right here are all processing. Okay, the reason I am showing you this is because if you have a pipeline that is taking some time to finish, okay, I'll go here, you will see that it will take a longer time the more pipelines are submitted. And especially if these pipelines include large data sets, use large databases as reference. For example, the size of genome is very important, right? So if we use mapping onto the human genome as opposed to mapping on the coronavirus genome, there's a huge difference in size. Coronavirus is only thousands, thousands of almost 30,000 nucleotides long. The human genome is 3 billion base pairs, or 3 billion nucleotides long. So you see the difference in mapping, because for each read, which is the same size, maybe anywhere between 30 and 300, but typically around 100 and 150 base pairs long, you have to find the best place to map that genome. So the ability to run these pipelines in parallel is limited if you are going to start many jobs. Now, when I say many jobs, specifically what I mean, I'll give you an example, and don't want to point out, anybody in kind of a negative way, but I do want to point out, for example, Arwa here is processing 1, 2, 3, 4, 5 pipelines. And I understand that they are different, but for example, if you are using an RSEM pipeline, that RSEM pipeline is going to take time. Because it quantifies expression levels, and here I understand that you're looking at macrophages, so we can look at exactly what is the reference genome and things like that. But you can see that the more pipelines you start running, and I know that some of you, because it was taking a longer time, you might be thinking, oh, okay, well, I'm not going to wait for this pipeline to finish. I'm just going to add another pipeline, and maybe that will finish faster. So that's not going to happen, right? So I just want to, ask you that if you want to be considerate with some of the other people that are also using the pipelines right now, make sure that you actually take the time to finish a pipeline that you've started. and only then move on to the next project so that you can allow others to complete their pipelines in time, right? So many people are starting to work on their independent projects, and that's an important consideration, I think, when you plan your project, to plan it so that you're not trying out things. You already know how you're going to get to the answer that you're looking for based on a pipeline that you've tried before and that you understand by learning from the courses that we have what the pipeline will give you. Okay, so are there any questions? So I know that there have been a few questions. The other question actually that I did want to talk about briefly is factor regression analysis. I saw that some people were struggling with factor regression analysis. So briefly, let me show you how factor regression analysis section works so that it's clear to everyone. In factor regression analysis, one of the things that you will not see is an input to any files, right? So you might be surprised in saying, okay, maybe something is technically wrong. That's actually not true. Why is that not true? When you click on Start here, and you go to factor regression analysis, right? And here, by the way, you have specific options for quantile normalization or normalization per replicate group, right? Which are important settings when you are trying to compare samples using them as replicates, right? Because in each group, the more samples that you have, the more high quality will be your analysis, given that each group has some variability that is comparable. So normalization is going to be here important. And then you have to set up these groups. So the reason why you don't just upload the data and then go through this process is actually to help you first organize how many samples are you going to use and how many factors you might have, how many levels for each factor will you have, and then you will be guided to upload files to add factors into groups. Okay, so prepare the file, or if you're going through Transcriptomics 2, and that's where you're learning about factor regression analysis, take the file from there, then set it up according to what types of samples and groups and levels you have, and then upload your files, and you'll be able to drag and drop them into appropriate places. So when you don't see an upload button at the beginning of the pipeline, I know it's counterintuitive, but don't be surprised in the factor regression analysis. Okay, are there any other questions that you might have had over the weekend when we had some time to go through all of these things that you would like me to address here in this session? So if you have any questions, you can post them in chat. But I hope it clarifies about speed of processing of the pipelines and factor regression analysis and just trying things many, many times to find out how it works. So if you want to read about factor regression analysis, that's in Transcriptomics 2. And if you want to understand why these basic RNA-seq processing pipelines take much longer, then go through Transcriptomics 1. It really explains at some level of detail how each step works and what are the complexities around the analysis that those steps represent. Okay, so if there are no more questions, let me continue with the program overview. We did some literature review, and I hope that okay, so Benjamin is asking, what marks the completion of this workshop? Yes, I will get to that towards the end. The objective for the program itself, I think when you say workshop, you mean the whole program, which had all of these sessions. So, okay, let me address that in greater detail. So if you go to this page, right, for this program, SARS-CoV-2 genomic data analysis, you can see that we have one more session left on the 10th of July, and that is going to be related to vaccine and drug design. Really, the objective is to submit a project proposal and get feedback from our team and from our external advisors on the project so that you can come out with a presentation. I'll talk about that in a little bit, how exactly to get there. But our hope is that before the 10th of July, you will be able to submit the project proposal. And Adekunle, in a second, I will go over how to edit your project proposal as well. And after you submit it, to get that feedback and address the feedback, right? So whenever you have a research project, it is basically an idea or a hypothesis that you have thought of, that you have explored and found answers for in data, and you were able to present it to your community of other researchers like yourself that are able to make sure that the analysis that you've done, the data that you have used, and the conclusions that you have come to are rigorous and indeed they are reproducible, and they are representative of some kind of a fact, right? So that ability to find facts and build our knowledge about COVID-19 and about this pandemic in general will help our community to grow in our ability to understand and accumulate knowledge around what's going on. So that's really the objective of this research program, is to help you get immersed into that research, data-driven research mindset, find the appropriate tools and apply them to the right kinds of data sets that we reviewed and that you've probably seen, and to be able to develop a project proposal and present it in front of your peers, getting the feedback and adding to that new information. Okay, so did that answer your question, Benjamin? Okay, so Adekunle, how to edit our project submission after initial submission. So BIPSA was very helpful to post some of these links. The project submission form, right? We have here two programs combined, one from Louisiana Biomedical Research Network and one just online. So you are able to submit the project in a form, and that form is dynamic. So actually every time you go back to that form, you'll be able to see what you've submitted. But we also extract the information from these forms, and we assemble them together into these project overviews. So we're going to go today through some of the submissions, and you'll see kind of where it is. But once we have a better sense of what each person is proposing to do, and identify strengths and weaknesses of each proposal, we'll share with you this full document so that everybody can take a look, potentially to be able to contribute to a project that somebody else started, or develop your own ideas based on some of these ideas that others present. Okay, so Arwa is asking, will we retain access to the TBioInfo platform after the educational program is over? If you wish to, there are options for licensing and using it for either research or to complete your educational project, and we'll discuss some of those options. And you can also reach out to BIPSA, who's on here, or email marketing@pine.bio. Right now, the objective is to allow you to use the platform to get to a project submission, and most importantly, to try and do that in a guided way so that you can get results and plan accordingly. So, the answer is yes and no. Yes, you can if you request, and we see that what's the purpose, and we have different options available. And no, it's not just going to be a continuous access after the program is over completely. Okay, so did I answer your question, Benjamin and Adekunle? Okay, great. So here, Adekunle, again, if you go to project submission, you'll see that form and you can edit it there. Okay, so we did focus a lot on reviewing some of these publications, and I hope that you were able to see that there are review publications that talk about methods and kind of general trends and different findings. You probably saw some technical examples that, for example, compare different pipelines and different methods and discuss settings and what are the outputs and things like that. And then there are actual research and discovery publications where something novel, something new is being found and discussed in the context of whatever data. So those are important types of projects that you can also present here. Not every research publication has to find something completely new. You can confirm something that somebody else has already found in a different study. You can expand upon something that somebody else studied, or you can actually just do a technical comparison and present your results, something that might be interesting to other users of the different pipelines or the different data sets. For example, many publications focus on identifying gaps in data to help generate new data to address those questions. Many publications speak about limitations of statistical methods or computational methods to give us comprehensive answers to some questions that we have. And other review papers are also important as a way for others to know about a topic and understand how different methods and different data sets fit into that topic. So I think all of these directions are valid if you want to incorporate them into your project. But what's important is to be able to also read through these review papers and projects and understand the research findings in the context of data and methods that they have used. Okay, so I'm just going to briefly review the sessions that we had. We talked about sequencing of viral genomes. We also talked about finding those sequences in different hosts and what can that mean in terms of the differences in those sequences. And to study the differences, we can really use a database of annotated sequences. And so, obviously, someone has to go in and annotate the different sequences and organize them into a database. And that is another major component of bioinformatics. It's not just the computational analysis, it's also developing these kinds of resources that others can use for reference, and they enable many different aspects of research using bioinformatics. We talked about statistical or computational methods, for example, a method of comparing sequences by what's called multiple sequence analysis to find areas of similarity or dissimilarity between different strains of viruses or different genomes. We talked about how to find those genomes and how to identify genomes in terms of their association with specific phenotypes. We also talked about phylogenetic or evolutionary analysis that allows us to see differences in sequences associated with mutations or variants in each one of those genomic sequences, and then building up an evolutionary sequence of origin and who came from whom, right? So a common origin between multiple sequences. We did work on some practical examples and ran some comparisons, and so we hope that you learn from some of the example data sets that we've prepared, how to organize the data, how to label your data, how to then make that pipeline more robust in terms of what kinds of comparisons by eliminating some of the outliers, some of the bad sequences that contain unknown characters or unknown nucleotide and amino acid sequences, and overall of bad quality. So for example, complete genomic sequences and incomplete genomic sequences, or different numbers of samples and groups. We also talked about how we can modify this pipeline to adapt to different types of questions and answers that we might have. And the purpose of this review is to really see where we are in terms of the program and group as a whole, because we're approaching the end of this program. And so as we are approaching the end of this program, I kind of wanted to make it also a little bit open-ended, and obviously we're going to start talking about the projects here in just a second. But before we do that, did anyone have any particular, so I wanted to mention that, if you do have any particular feedback about the program that could help us improve, develop new materials, and help you get the most out of the tools and the data sets that we discussed in the context of these programs, including, by the way, what other topics that we did not have time to cover in this program but are interesting to you. We're always waiting to hear for your feedback, whether positive or negative. We hope that it's constructive so we can learn and improve in the future. But I do want to mention that that will be an email that you will receive from Bipsa, where she will ask you to give us some feedback and kind of rate the different elements of the program. And this is an important part of also us understanding what are the best practices and the best solutions to allowing people like yourselves to learn bioinformatics, and what is it that you can do after a project or a program like this. We will have a session for vaccine and drug design. I believe that many of the aspects that we covered in the program were leading up to that point where we would be able to understand better what goes into vaccine design, what goes into drug design, what goes into drug repurposing. And we hear a lot about this in the news, but kind of linking it back to data and the challenges around the COVID-19 pandemic and coronaviruses in general. So we are still looking for a guest to contribute to this lecture. We already have a few candidates that we've reached out to and they've expressed interest, but we're going to hope to combine our kind of insights from that domain with an expert who has been doing research in that domain for a long time that would provide you with an expert kind of perspective on some of these challenges. So hopefully, as we approach that time, June 10th, you will be also thinking about some of the questions that you might have and preparing those questions in a format that somebody who's an expert in the field can really answer those. Okay, so let's briefly talk about these independent projects. So what is the objective of these independent projects? So as I mentioned, the objective is to find a problem, a specific focus problem that you can focus on, and from that problem statement, collect raw data that you need to answer your problem, to define the kind of context, to define specific questions, and then to be able to find relevant data that we know and we can evaluate that that data can actually answer the question that you're posing. After that, you need to select the methods to process the data and prepare and organize it for analysis. Do exploratory analysis. Exploratory analysis in statistics is very important because what statistics allows us to do is to take a sample from all the data that we want to study. For example, if we want to study all the people in the whole world and we want to describe some biological condition, for example, response to COVID infection, we are taking samples, a small number of samples, to make some inferences about the larger population. And so selecting the right data and focusing on the right questions, understanding the variability of whatever that data represents is an important part of preparing data that will provide us with some clear insights into those questions. Maybe not conclusive, but some insights. Exploratory data analysis helps us eliminate issues that will lead to improper conclusions or wrong inference. So it really allows us to describe the data that we have, statistically speaking, and understand whether we have some outliers or trends that are not expected, or issues that will even prevent us from doing proper statistical analysis of the data. Then we speak about the in-depth analysis, and in-depth analysis includes processing, maybe statistical analysis, or even some computational analysis like machine learning, but ultimately it also includes interpretation. Now, interpretation takes a long time, and it really requires an in-depth understanding of the problem in its context, right? So we are not expecting you to necessarily achieve a complete research project in such a short time frame. We're not expecting you to make some groundbreaking discoveries, even though we're not, thinking that you can't or it's not possible. But what we do want to mention here is that, your in-depth analysis, once you have described what you have done and applied to which data set, the results of your analysis could be as simple as, I understood what's in the data, I found some interesting trends, and these are some indicators that these trends are, biologically relevant. So even if you find something that biologically speaking makes sense, right, it might not be, necessarily conclusive, or you might need to do a lot more analysis after this, and you realize that, that is still a result, right? The objective here is not to publish. The objective is to really be able to present what you have achieved in this program. And so the more of you that are, brave enough and ready to try, I think the better for all of us, because we learn from each other's mistakes, we understand the limitations. And so any review that is given in this program is not to say that, your analysis is bad or the topic that you've selected is bad. It's really to help you improve some of your analysis and make sure that, it is interesting and compelling, but it doesn't have to be, groundbreaking and exceptional. So that's what I'm trying to say. Okay, so when we want to frame the problem, right, the important part of it is to understand that there are many different directions that make this problem interesting. It could be basic research, it could be clinical applications, it could be biotechnology and pharmaceutical related applications. These different directions really allow us to select a topic of interest and kind of understand how they are addressed in very different ways in terms of these different problem statements. Collecting the data, we talked about at least a few different resources, including the NCBI SRA, NCBI virus, even sets like those included in visualization tools like you see here, and also GIS Aid, which is a portal that I know last time we discussed, and many of you maybe have tried logging in and registering. So if you are having any issues with that, we're also happy to help. And so this week, we're actually going to be going through some of those challenges to make sure that you are able to find and download the exact data sets that you're looking for, if they exist and if that is possible, of course. So again, how to submit your project proposal for initial review. You go to the project submission form, and the project submission form is dynamic. So you can change any element of this project submission, and we will see that you have made the changes. So we actually get an email notification when you make those changes. Last time we reviewed one of the projects, and again, I hope that the review was helpful. It is not meant to be criticism of what you're thinking of doing. I think that project idea was interesting, but it is important that once you have an interesting project idea, that you actually fulfill the other requirements, that you are able, for example, to find the right data that will describe the problem and give you a source of answers for your questions. And also the ability to explain how you will actually do the analysis. So some of the previous projects that the students and faculty and researchers have done were really impressive to us. Again, many of the projects that you see right now described on our project repository came from these student and faculty collaborations. Many of them end up going to different conferences and presentations, and now it's a great time to do online learning, because also all of these conferences are also online. So if you really have an interesting result, I think it's worthwhile to making it as complete as possible so that you can explore those options. Now, what goes into the final result here? You need to have a title. You need to have a description, an introduction, basically, to what the context is. You need to describe the methods of analysis, results. So what did you find? What kinds of patterns, what kind of interesting genes, mutations, or, etc. you were able to find. And then to be able to speak about the conclusions, right? So those are the 4 elements that you need to think about: introduction, methods, results, and conclusions. And so this could be a project that you work on independently, or it could be a project that you work with others on, and each person can focus on one of these elements. For example, one person can focus on the introduction, giving literature review and kind of the biological context. Some of you might be more versatile in terms of analysis methods, and you can run multiple analyses and extract the right kinds of visualization and outputs. Somebody might be focused on interpreting the results and writing the conclusions, right? So those are some of the aspects to think about as you through these projects. Yeah, did you want to say something? I heard somebody speak. Okay. Okay, so now I wanted to kind of go through some of the project submissions that we have received. We also already went through Ramasha Gupta's project. Benjamin, you also submitted a project, and so I wanted to find out, did you want to actually present some of this and speak about it yourself, or would you like me to do it for you? Benjamin? Yeah. Do you want to speak about your project?
Yeah, maybe you can do it for me.
Okay, no problem. So how we typically do these is we say just a few words about who you are and why you chose the topic that you chose. That's not really included in the project description, but do you want to just mention briefly a little bit about yourself, who you are, what were you interested in?
Maybe, yeah, sure. I'm Benjamin. I'm doing diagnostic testing, molecular testing for several kind of targets. And the thing is, right now I want to move towards research. So I'm exploring this bioinformatics, and I have this recent employment with A-Star, which is actually a biomedical research facility in Singapore. They're doing transcriptomics, so that's where I'm trying to get some insights before I join the company in August. Yeah, so that is pretty much why I'm trying to.
Okay, great, thank you. So we don't have a title yet, and we don't have a presentation, but as you've heard, the interest here is to gain some experience and really see what these data sets are all about. So let's see what Benjamin submitted. So he mentioned that COVID-19 often starts with non-specific upper respiratory tract symptoms, making it difficult to distinguish from those of other diseases. In particular, influenza virus infections may present similar symptoms as those of COVID-19. The current lack of clinical knowledge about COVID-19 might therefore lead to bias and misdiagnoses, and in cases of coexisting infections. We would want to understand the different responses from the body to COVID-19, influenza virus infection, and co-infection. Okay, so briefly about the problem statement. I think it's worded well. There are some just minor changes that I would suggest, but otherwise I think it's very clear, right? You've identified what the problem statement is. You've identified what is the specific type of comparison that you think will address that issue. Problem summary. Influenza spreads globally annually with significant pediatric and adult attack rates and considerable morbidity, mortality, and the exacerbation of extant chronic diseases. In the northern and southern hemispheres, outbreaks occur mainly in their respective winter seasons. Influenza vaccination is available but only partially effective. In the absence of a vaccine in winter, novel coronavirus COVID-19 will also circulate in parallel with seasonal influenza. Thus far, it appears that with the current strains of these two viruses, the clinical outcome of co-infection is not significantly worse than infection with COVID-19 alone. Similarly, however, several strains of influenza circulate, including strains that are still to come. Similarly, COVID-19 has several strains with probably more to come, and to better prepare ourselves by understanding the body's biological response to the virus. Okay, so before putting the project viability, I think that's a good idea, just to check if this is a good direction. So here we have influenza vaccination in the COVID-19 era. For me, I think the overall idea is great, and I even know of some specific data sets that compare, let's say, chronic conditions like asthma, different types of upper respiratory infections. I'm not sure that it's specifically flu, but other viruses and COVID-19. So, in that sense, it's very clear to me what you're trying to do. What complicates this is the component of vaccination, right? You mention it through some of the project description, but that doesn't seem to be a specific focus of your problem statement, right? So I would just differentiate. If you want to specifically focus on the aspect of vaccination, so a vaccine, an attenuated vaccine, is kind of like a virus in itself, right? It should cause some immune response, but that immune response is more to train the immune system. Typically, an influenza vaccine causes very mild symptoms, right? So maybe the question, if you want to make it more specific to that vaccination, is how does COVID-19 infection or immune response to COVID-19 is modulated by an influenza vaccine, right? That is a separate question. I think a very interesting one, because we have influenza vaccines and all kinds of other vaccines, right? But then it, I think, will be a little bit more challenging to find the data, but it's an interesting question. And we can explore whether those types of data sets are available. And if you want to focus exclusively on influenza, then also, you might want to explore further, the current developments of coronavirus vaccine, the current trends of influenza vaccines. There's also multiple candidates for vaccines in influenza that are either already in use or planned to be in use in the future. So I think if you want to focus on the vaccine, a little bit more about vaccine trends in these two, in respect to these two viruses, is going to be interesting to see in your project description. But I think overall the project is, a great topic, and interesting. What you think might be the next step? What data would you use? What type of analysis would you plan for this kind of data, etc.? So, Benjamin, did you want to comment on any of that?
I think the vaccine part will be biting off too much. I can chew. So I'm like maybe that will be like a secondary kind. But my primary focus is more of like the body's response to the three variations. It might be because the influenza virus upon infection have the same symptoms as the COVID. But the thing is, molecular response is different. It's different. So I was like thinking, how maybe I can show it in the sense like each individual and the combination, a hybrid of COVID with influenza. What's something like meta genomics in a sense, is it?
Yeah. So I see what you're saying. So you want to focus on response to.
Yeah, that's my main focus.
Okay. Yeah, so I think that makes it, more straightforward, right? That is clearly explained in your project description. And,
So the thing is
here,
actually a reference, because I put it as a reference to the co-infection and how the vaccine is actually being produced. So I put that as a reference, but my main focus is on the body response. Yes.
Mhm.
Yeah.
Okay, well, let's briefly take a look in terms of data sets. This is the data set that I was referring to, even though you also have this. The problem here, I believe that it doesn't include, let's see, what types of samples does it actually have? It has a lot of samples. And I'll post this, maybe the previous one, so you can explore this data set. If you click here on SRA experiments, well, in fact, we don't even have to go there. You can just read about it right here, right? So here you can see, okay, in-depth analysis of the host response to SARS-CoV-2 as it compares to other respiratory infections, cell and animal models of SARS-CoV-2 infection. In addition to transcriptional profiling of COVID-19 lung biopsy consistently revealed a unique and inappropriate inflammatory response defined by elevated chemokine expression in the absence of type 1 and type 3 interferons. Identification of a muted transcriptional response to SARS-CoV-2 supports a model in which initial failure to rapidly respond to infection results in prolonged viral replication and an influx of pro-inflammatory cells that induce alveolar damage and manifest in COVID-19 lung pathobiology. So what types of infection did they check and what types of tissue? Lung epithelium, mock treated infected with SARS-CoV-2, IAV, so this is H1N1. So this is influenza avian virus that lacks the NS1 protein. So this comparison right here is not going to be necessarily, your typical influenza strains, but I think they're specifically designed to show how it is different from influenza. So if you take this, the only thing that I would highlight here, influenza A, right? So influenza A virus is, again, you want to make sure that you think about how long will it take you to do this analysis, what are the expected results of each pipeline that you want to run, and how do you actually communicate, going from maybe a little bit better defining what your question will be, because you have multiple tissues, multiple animal models, and ultimately many samples, right? You're not going to have time to process all of them and find everything there is to know about the differences, right? Maybe just focus on one particular aspect, one type of tissues, and then build it from there. All right, so great. For the purposes of time, let's keep going through. Arwa, would you like to speak about your project?
Hello. Good morning. Hello, everyone. Sir, please show my project page. Sir, even if you explain it, I'm fine with it.
Okay, could you start with just a few words about yourself and what?
I will. Hello, everyone. I am Arwa Bora from All India Institute of Medical Sciences, Bhopal. I'm a third-year medical student, and I'm very interested in the field of cancer research and immunology. I think that bioinformatics is the most feasible way of carrying out research, because clinical research takes a lot of time and does not change anything potentially. So that's why I logged in here, and that's all about it.
Okay, great. Thank you very much, Arwa. Let's go through this project description. We still don't have the title of the project or the presentation. Those would be the next things to start working on. But let's according to PICO format, research problem upregulated genes in PBMC. Okay, so PBMC, what does that stand
Hello, PBMC are peripheral blood monocytes. So in simple words, they are macrophages present in the blood. Peripheral blood mononuclear cells.
Okay.
Yeah.
So, remember that many of us are not going to be familiar with some of the terminology, so please make sure you clarify. Okay, so according to the format, research problem, right? Upregulated genes in PBM prevention, drug interaction, comparator infected versus control, out drug gene interaction, research question, which druggable genes are upregulated in PBMC in a SARS-CoV-2 infected in SARS-CoV-2 infected patient? Okay, so this right here should be your problem statement. Okay, so this should be your project statement. But you would want to add here some background, right? So what are PBMCs? Why are they important for COVID?
Hello. Yeah,
Yes.
actually, we cannot control the virus which is coming from outside because every time the virus comes, it might come with a changed genome, and we have a lot of phylogenies for that. But we can control what the immune system does. So if we look at our macrophages, and if we look at what genes or what molecular pathways are being upregulated in our macrophages, and if we downregulate those pathways with some immunomodulatory agents, so actually the death in COVID-19 patients is because of the immune toxicity and not because of the virus itself. The virus has less virulence and it's not that strong of a virus, but our immune system actually comes into the lung, and the pneumonia is the problem. So that's why if we control our immune system, so that way we can actually control the infection pretty early.
Okay, so that's perfect. You could add that into your project statement. That will all give us, a better sense of why we're talking about PBMCs, and what is the direction. So I think just add that into your project submission. Let's see project summary. So one, RNA-seq analysis to get tables of expression and isoform expression, and then factor regression analysis and principal component analysis. Search the genes that encode proteins and for which potent chemical synthetic inhibitors are available based on drug bank. Next, subject to functional annotation and construct eye diagram. Okay, so let's look here, because what we need for both factor regression analysis and any kind of comparison is to understand what kinds of data are we planning to use. So here we have single cell atlas of the peripheral immune response to COVID-19. Again, I am kind of familiar with this data set. I would, try to specify which specific data sets from this collection you plan to use, because there are specific types of controls, right? So here, for example, you have 7 patients hospitalized for COVID-19, 6 healthy controls. So you have a, severe infection if they're hospitalized, right? And then you have some health. Once you do that kind of comparison, why did you choose, for example, factor regression analysis and not just differential gene expression?
Okay. First of all, one thing I think I don't know if it's correct or not, but I intend to use one of the controls as the reference genome, healthy controls as reference genome, and one of the infected patients as the infected, and will I plot against the infected patients against the reference genome and construct kind of phylogenies if possible. Now, we could have used differential gene expression also, and we will be definitely using differential gene expression, but the reason to use factor analysis and then further principal component analysis is to try to remove some further factors which are contributing to it, and to as in specify which particular immunomodulatory pathway is more involved. For example, at the end, all pathways may be a component of any tyrosine kinase inhibitor maybe, or KTOR. But if in PCA they differentiate between KTOR and tyrosine kinase inhibitor, this makes us go a long way in choosing the drugs, because the classification of drugs becomes entirely different. So PCA just to find the most appropriate pathway to target, maybe.
Okay, so thank you for clarifying. Let me briefly summarize, some of my feedback initially, and if you hear that it has any questions, you can add those in the chat. Briefly speaking, I'm just going to, sorry, for now, we have the healthy patients and the infected patients with a severe infection. Their genomes don't change, right? So the human genome does not change as a result of a viral infection. So we do not need to compare genomic sequences from these patients. What we want to compare are the expression of genes that are upregulated or downregulated as a result of an infection. Now, you probably meant transcriptome, right? So we can assume, but I think that's a little bit of a problematic statement, that the healthy controls represent a healthy transcriptome. And the infected people here had that similar, healthy transcriptome before they got infected or before they got to a severe infection. In some sense, that is true, because we want to identify, what is different about the infected people as opposed to the non-infected people, right? What I would just look into is a little bit of a better understanding of who these people are, right? So as we've heard many times, people with comorbidities, issues of age, even genetic background, or gender or sex might play a role in many of these types of comparisons. So those would be some of the control variables that I would try and look into as you explore the specific data sets. So you can go into the SRA experiments, right? So we know it is 6 plus 7. And here you can go to the run selector, and you can actually download the metadata. So when you explain your project, it would be great for you to even prepare some kind of a table or a visual chart that says, okay, I've selected these patients. So here you can see healthy control, patients with confirmed COVID-19, but you can actually download metadata, okay, and look inside if anything else is known about these patients. Okay, and the other thing is I would just work a little bit better on the terminology. So you can actually look at, how do you explain the specific comparisons and the anticipated results. So you did mention, changes in different pathways, but you also mentioned a lot of these different, pathways affected by drugs, which I think is great, but it's just important to maybe do a little bit more explanation about how these drugs work. And so I wouldn't go into, okay, let's take all of the drugs, right? All the possible drugs. Give us some specific examples of drugs that you think we should be thinking about. So for example, you say here, potent chemical synthetic inhibitors, right? Maybe suggest a couple or even just one that you think is most promising in terms of inflammation response, so that we can, just have a more clear focused direction to say that, we will focus on the findings of the different gene expression profiles to test whether common, inflammatory inhibition drugs are going to be effective for those kinds of patients, right?
Excuse me, actually the gambit of drugs can include from simple steroids like dexamethasone to some very specific drugs which can target one specific molecule in the pathway. So that's why I've tried to plot it. We'll first get the results from gene expression, and the genes which are expressed or are upregulated, transcriptome which gets upregulated, will try to find them in the drug bank or drug gene interaction database. So for like to begin with, I don't have any specific drug to target. It's not like as you say, it's an evidence based research. It's not the question based research kind of.
Okay, so you want to just explore. So maybe that's what you want to say again here in your project statement, right? So because you say which druggable genes are upregulated in PBMC and SARS-CoV-2 infected patients, right? But you're also asking which drugs, right? From, you know, from known, you know, maybe approved, right? From known and approved medications might be effective, right? So it makes, what I'm saying is that this makes your project much broader and will require a lot more downstream analysis if you want to include that second component, right? Because I can see you in a fairly short period of time doing the RNA-seq, finding differentially expressed genes, and even understanding which pathways are up and down regulated. I think that is feasible. Then necessarily performing, you know, the second part of it would be an additional component, you know, that will require time, right, for you to actually work through that. So it's just a matter of how much effort will that require to complete those steps. Okay, but thank you for sharing this project. You know, again, you can go into the submission form right here and just modify anything you want to change. And obviously, as you are making progress with your analysis, if you could please add a link to your presentation, even if it is in progress. So it doesn't have to be complete when you share it. It can be just a draft. We can start seeing what slides you have, what pipelines you're running, what results you're getting, that kind of thing.
Also, I'm not able to work with any of the SR experiments. Whenever I try to use the .csv format or NCBI run tables, it somehow does not. I believe that I do not have any access to use SRAs right now. So then how do I convert SRA to FASTA, like from the NCBI toolkit, or will I get the access to use the SRA formats like with the run table info?
No, everybody has access, so let me show you. So this is the CSV file that I downloaded from that study. If I go here, I'll go to RNA-seq. The important part is to know, so let's take a look at what's in the CSV. So this says that these are SRA FAST-A, FAST-Q, and here it should say if this is paired end or single end. Paired, okay. So let's change this to PE. Here we will use the human file upload. Instead of using upload files, you actually go to NCBI run upload right here, okay? And again, so
Actually,
one second. We do have to move on to another project. So let me just quickly go through this.
Okay, sorry.
Okay. Okay, so here you go to NCBI table upload, add files, and this is the CSV, choose, group selection. You see all of this?
Excuse me, now you actually got all the .sra formats now. My computer always shows 100%, but it will always show .csv. It will not process the files. I do try to see the paired ends and everything correct, like, but it never you know, goes the next step and shows
Okay, so
.sra.
again, make sure that you're uploading not here, not here, not here.
I have even tried to be vigilant with everything, but still it somehow
Okay, maybe we'll set up a different time then. Maybe you can send some screenshots and you know, we'll troubleshoot it. But as you can see, it works on my end, and there's no other setting, you know, there's no way to prohibit you from doing that. So that should be working. If there's some, you know, other issue, we can look into it. Okay?
Thank you.
All right. Okay, so again, thanks for that presentation. Let's go on. Ahmed Jafari, would you like to briefly speak about your project? Are you on?
I don't see him here either.
Okay, all right. Therese, are you around?
Yes, I'm around.
Okay,
Hello.
would you like to say a few things about your project?
Okay. I realize this is kind of, I think I oversimplified it, but I was just thinking that I wanted to do something doable. So I identified like a problem that I wanted to look at, which is the coagulopathy that was being seen in some severe cases of COVID-19 patients. And I looked at some literature, and the mechanism of which this occurs is not yet fully understood, although there's many reviews talking about how there's elevated other biomarkers such as D-dimer or fibrinogen levels in these patients, but the mechanism is kind of different when they look at COVID-19 patients. So I wanted to look at 3 particular genes that I identified that are part of the coagulation cascade. So those are the PLAT, PLAU, and the PAI1. And I just wanted to see how they are downregulated or upregulated in patients with an RNA-seq profile. And that might give some insights on the mechanism of how this coagulopathy occurs.
Okay, so I think, yeah, if we look at your problem statement, right, so transcriptomic analysis of plasminogen activators in bronchoalveolar lavage fluid of COVID-19 patients, I think, you know, what you described is fairly clear. Let's just briefly take a look at the data set here that you provide.
Yep.
So 10 samples were subjected to metagenomic next generation sequencing. And what types of data sets do we have here? The time, origin, genetic diversity, transmission dynamics, and evolutionary history of SARS-CoV-2 were analyzed based on the genomic data to provide understanding on the origin, transmission pathway, and evolutionary characteristics of SARS-CoV-2 outbreak. That's interesting.
I realized they might have focused on the viral genome in this particular study, but I was hoping that the human total RNA would still be there, the reads. Do you think that they would still be there?
Yeah, the reads are here, but you know, I only see 2 with bronchoalveolar lavage UID. So are you going to use only these 2 samples?
I did want to just limit my sample size to like these 2 things, because I was trying to find other studies that had bronchoalveolar lavage, but I couldn't find much. I did see earlier you flashed like a single cell study that was done, but I wasn't sure if what the tissue samples was for that one. But if
Right,
you
so
could point out,
right,
if you
the problem with trying to do a comparison between 2 samples is that you don't have any way to control for, you know, variation not associated with something, right? So if you, for example, it looks like all the samples here are infected, right? So it's not like
Right.
you have infected versus uninfected. You don't have a healthy control.
Right,
So,
that's correct. If you do have, you know, like a suggestion of some data that would be better to use for this, I think, yeah, that's the one.
Right. Here, they actually have a lot of different samples here, and I would suggest looking into it, because for COVID-19, so let's just, So right here, you have independent biological triplicates of transformed lung alveolar cells. You have, what else do we have for alveolar? Okay, so that's what you have here. As you saw, there is another one. Well, this is COVID-19 clinical isolates. I think there's maybe another data set that I've seen on the single cell, right? It's a different one, not this one. But that single cell also has a lot of data sets that you can explore. So, you know, I think typically you would have at least 2 samples per condition. If you have 3, that is better. You can see triplicates here. Why? Because if 2 have, you know, differences, which one do you choose, right? Which one is the correct one? The third one would be the tiebreaker. So that's typically your design for a differential gene analysis, but I think overall the project idea makes sense. If you want to refine some of the, you know, specifics about the data sets, and also honestly, what's not very clear to me is why should we only focus on these 3 genes? Would you not want to find all the differentially expressed genes, potentially look at some, you know, upstream, downstream regulation based on those genes, right? So just because those genes are involved, does that mean that you should expect them to be very differentially expressed?
I guess I just wanted to narrow down on those 3 genes, but I did think about, because I saw a lot of studies already doing looking at expression of immune, like cytokines, but I didn't want to go in that direction. I'm more of interested in the hemostatic biomarkers. So I guess I just wanted to narrow down on that. But if you think that if it's doable to look at other genes too, then I'd be open to working on that too.
Okay, great. So let's first find some data sets, right? And if we can find the data sets and then see, you know, what the, again, it would be great if you could also describe the specific analysis that you plan. So, you know, here are the samples.
I just wanted to do a differential expression, but I still need to review my transcriptomics. Yeah, I'll refine the methodology for that.
Okay, excellent. So those were the projects that were submitted. So I think all of these projects were really interesting in terms of the topics that were highlighted, right? I think we really see the way some of these methods and data sets become useful when we start planning these types of analyses. In the next few sessions, what we will do is briefly review some of the additional projects that are going to be submitted. I do want to mention that, you know, many of you are asking about how to continue and get access and, you know, what will happen after this, or you might be interested in other elements of this kind of data analysis. So to summarize, I think that, you know, please continue to submit these projects. We'll give you feedback and we'll do some more reviews and sessions like this. If you are interested in becoming, you know, a mentor, joining another program, please reach out to Bipsa, and she has some additional programs. Actually, one of them is starting today. Later in the afternoon, we will start our Omics 2020 data science program. So anybody that is interested in R and Python programming and machine learning and data visualization will have a program specific to that. And I do want to mention that, you know, we will have another session focused on vaccine and drug design. And so if you have any questions or you're working on a project related to that, please prepare those questions. You can put them on the forum to make sure that they are getting addressed. If there are others, so Lila Rodriguez, please reach out to Bipsa at, so her email is marketing@pine.bio. Yes, Adekunle, you just edit your and click on submit again. So that should work. We should get an email based on your changes. So again, the specifics of the projects that you've seen today are going to continue and evolve over time, hopefully. And any one of you that is interested in these topics that we talked about today, you can go to the profiles. That's why I actually wanted some of these people that presented today to speak briefly about their background and who they are. So we heard from Arwa, we heard from Therese, and from Benjamin today. So if any one of you is interested to join their projects, you can find their profiles and directly message them, or we can start a discussion on the forum about who might want to join who. For those of you that did update your profiles, if you could, Therese, so if you want to add, link to your social media like LinkedIn or something like that, if you want people to reach out to you, or ResearchGate or something like that, you can do that here. We're really hoping that as a result of this program, you'd be able to not only work on a project. We understand that it's time consuming and does take a lot of focus. So if you don't have that time, maybe that's a second solution. You can reach out to others that might be interested to work on something, don't really have a defined project in mind, but still want to contribute. So again, thank you for those that presented today. Adekunle, if you want to reach out to her, again, this is her profile right here. And you guys can communicate directly, or if you want to get help with some of this, we do have here Bipsa and Mohit that can also help connect you and make an introduction if you're interested. Hopefully we will have a few more project ideas submitted and some of these refined for our next time. And we'll certainly do the best we can to address any issues that come, like finding the right data set, or selecting the right type of analysis, or thinking whether that is achievable or feasible in a short period of time, depending on what you have, etc. Thank you for joining today. Sorry for the technical issues at the beginning of the session, but we will have the recording available on the forum, and we look forward to seeing you again next time.